Today, we're going to take a very high-level quick peek into prescription drugs that are being investigated for off-label use as longevity enhancers. This is a preview of pharmacological tools that may influence fundamental aging biology, a direct look at what prescription drugs are already reshaping how we think about health span and lifespan, and what still needs to be proven. If you're a clinician, researcher, systems level thinker, or even hobbyist in aging biology, this is for you. If you're interested in learning more, leave me a comment and we can go and do deep dives into each of these. So today, we'll analyze 7 drugs and categories of prescription drugs that have strong mechanistic rationale, broad system level effects, or early human evidence suggesting they might alter the trajectory of aging. Specifically, Metformin, Rapamycin, PDE5 inhibitors, PDE9 inhibitors, PCSK9 inhibitors, Acarbose, and SGLT2 inhibitors. These aren't just treatments for metabolic or cardiovascular disease. In the right context, they may be tools to extend health span, but we're not interested in speculation. We're here to look at pathways, trials, evidence quality, and what actually moves the needle. Let's warm up with Metformin, AMPK activator, mTOR indirect suppressor, improves insulin sensitivity, reduces hepatic glucose production, alters the gut microbiome, reduces oxidative stress, modulates autophagy. In diabetics, we've seen lower cancer incidence, lower cardiovascular disease, reduced cognitive decline, and lower all-cause mortality. Sometimes even better than non-diabetics not on metformin. But that's epidemiology. Dr. Nir Barzilai's TAME trial is designed to test this properly. Does metformin delay the onset of multiple age-related diseases in non-diabetics? Until that reads out, everything else is signal, not proof. Now, rapamycin hits harder. This is a direct mTOR complex 1 inhibitor when used intermittently. It suppresses growth signaling, ramps up autophagy, slows protein synthesis, and enhances cellular repair. In mice, rapamycin extends lifespan even when started late into adulthood. It's the strongest geroprotector we have in animal data, having extended lifespan in every animal species studied. In humans, low-dose intermittent rapamycin improves immune function, vaccine response, and may slow some aspects of age-related decline. Pearl trial data shows lean mass improvements, quality of life gains, and no spike in adverse events. Weekly dosing looks safe. High dose daily is not. If you're prescribing or using rapamycin, you need to be precise. PDE5 inhibitors are next. These aren't just erectile dysfunction medications. They preserve nitric oxide signaling by preventing cGMP breakdown. That improves endothelial function, blood flow, possible neurovascular health. Observational studies show lower cardiovascular events, lower all-cause mortality, even reduced dementia incidence in chronic users. No randomized controlled trials yet, but the vascular mechanism is real. These drugs are cheap, safe, and mechanistically relevant. Especially in older men with vascular risk, PDE5 inhibitors like Cialis and Viagra are worth a serious look. PDE9 inhibitors are newer. The mechanism is similar but works on a different pool of cyclic guanosine monophosphate or CGMP, the one linked to natriuretic peptide signaling, mostly in heart and brain tissue. Early trials show promise in heart failure. No longevity data yet. But if they reduce cardiac fibrosis and improve perfusion in heart failure with preserved ejection fraction patients, they could be a niche cardiogeroprotective agent. PCSK9 inhibitors are the most direct tools we have for reducing atherosclerotic burden. They reduce LDL cholesterol by up to 60% beyond statins. If you believe cardiovascular disease is an aging pathology, and you should, then PCSK9 inhibition is a frontline intervention. Evolocumab and lirocumab reduce major cardiovascular events. All-cause mortality trends downward in trials. Safety looks clean. Inclisiran, the siRNA version, only needs to be dosed twice a year. This is aging medicine through the lens of lipid control, targeted, reactive, and already in clinical use. Acarbose is a gut glucose absorption inhibitor. It blunts postprandial glucose and insulin spikes. In the NIA's ITP study, it extended mouse lifespan by 17% in males. In humans, the STOP NIDDM trial showed reduced new onset hypertension and fewer cardiovascular events. The larger ACE trial didn't confirm that. GI side effects limit adherence, but mechanistically it mimics some caloric restriction effects. While it may not be a first-line tool for many people, it's certainly relevant as a concept. Glycemic modulation matters for aging. SGLT2 inhibitors might be the most underestimated drugs in this conversation. They were originally built to lower glucose through glycosuria, but they turned out to reduce heart failure hospitalizations, protect kidneys, reduce cardiovascular mortality, even in non-diabetics. Mechanistically, they lower insulin, reduce weight, promote mild ketogenesis, and reduce systemic inflammation. They also improve cardiac metabolism by shifting energy substrates. The net result? Slower decline of critical organ systems. In terms of human data, not mice, not proxies, SGLT2 inhibitors have the strongest evidence on this list for extending health span in real people with real disease. And that puts them firmly in the category of drugs that deserve attention in aging research. Mechanistically, there's overlap across many of these. Metformin, Rapamycin, Acarbose, and SGLT2s all modulate nutrient sensing. PDE5, PDE9, and PCSK9 inhibitors modulate vascular aging and perfusion. What's missing from this list? Senolytics. None of these eliminate senescent cells directly, and we're not hitting stem cell exhaustion or telomere attrition either. These are key aging pathways that still need tools. That's the next phase. Agents that clear senescent cells, reprogram epigenetics, or preserve stem cell pools. Right now, those are in early stage trials or under development, but they're coming. Meanwhile, supplements like fisetin and alpha-ketoglutarate are being tested as potential senolytics or rejuvenators and should be watched closely. To that point, the Novo's core formula, which contains both of these substances and many more, has a growing list of studies in vitro and in vivo that are validating its unique impact and efficacy as a longevity intervention. Ultimately, polytherapy is essential. You can't treat aging through a single node. AMPK, mTOR, insulin, nitric oxide signaling, LDL, CGMP, perfusion, inflammation, they all need attention. A well-structured pharmacologic longevity protocol will map intervention to individual risk. High LDL, PCSK9, insulin resistance, metformin or SGLT2, hyperfunction and inflammation, rapamycin, endothelial dysfunction, PDE5. This isn't kitchen sink biohacking. It's targeted molecular strategy. And to be clear, none of this replaces sleep, diet, physical activity, or stress management. These drugs aren't lifestyle substitutes. They're layered augmentations for people who already have the basics in place. If you don't have a solid foundation, these won't save you. But if you do, they might stretch that health span even further. The TAME study will push the regulatory needle. If it succeeds, it may open the door to aging as a composite endpoint for the FDA. But we don't have to wait for that to think critically. Many of these drugs are already prescribed for standard indications. The future is in optimizing them for prevention, earlier, smarter, and more strategically. Aging is modular. Our interventions should be too. Some drugs hit metabolism, some hit vasculature, some hit cellular stress. Stack them based on biology, adjust by biomarker. Monitor function, not just disease. This isn't about slowing time, it's about maintaining capacity. That's what these drugs are for. When deployed with precision, they can shift the curve. Not hypothetical, not futuristic, just underused. So until next time, age wisely and slow your age.